Dear students of semester 2 honors, today we will discuss about protein sorting and vesicular transport under the cell biology. Actually, we know that proteins are synthesized by ribosomes and afterward the proteins are sorted. Actually, proteins synthesized on free ribosomes either remain in the cytosol or are transported to the nucleus, mitochondria, chloroplasts, or peroxisomes. In contrast, proteins synthesized on membrane-bound ribosomes are translocated into the ER, that means endoplasmic reticulum, while their translation is in progress. They may be either retained within the ear or transported to the Golgi apparatus and from there to lysosomes, which is termed as GERL system, or the plasma membrane or the cell exterior via secretory vesicles. So this slide shows the overview of this protein sorting we have already discussed. Next. In the next slide, the topology of secretory pathway is shown. Actually, if we want to know the topology of secretory pathway, then we can say that the lumens of the ER and Golgi are topologically equivalent to the exterior of the cell. Consequently, those portions of polypeptide chains that are translated or translocated into the ear are exposed on the cell surface following transport to the plasma membrane. So, overall, the secretory pathway of any secretory protein is ER to Golgi to the cell plasma membrane and then to the exterior. This is targeting secretory proteins to the ER from ribosome. Actually, we know that this is the first step of GERL system. As the signal sequence emerges from the ribosome, it is recognized and bound by a particular protein that is called signal recognition particle or SRP. The SRP then escorts the complex to the ER membrane where it binds to the SRP receptor. Then the SRP is released for the next cycle and the ribosome binds to the membrane translocation complex and the signal sequence is inserted into a membrane channel. Translation resumes and the growing polypeptide chain is translocated across the membrane. Cleavage of the signal sequence by the signal peptidase releases the polypeptide into the lumen of the ER. This is insertion of a membrane protein. Actually, there are some proteins which should be inserted in the Golgi, in the ER membrane, How this phenomenon does occur? Insertion of a membrane protein with a cleavable signal sequence. Actually, the signal sequence is cleaved as the polypeptide chain crosses the membrane. So the amino terminus of the polypeptide chain is exposed in the ER lumen. However, translocation of the polypeptide chain across this membrane is halted by a stop transfer sequence that anchors the protein in the membrane. The ribosome is released then from the membrane and continued translation results in a membrane spanning protein with its C terminus on the cytosolic side. When the protein goes in the lumen of the ER, then some modifications occur within that lumen. Like the polypeptides fold into their three-dimensional conformations within the ER, assisted by the molecular chaperones that facilitate the folding of the polypeptide chains. For example, one of the major proteins within the ER lumen is a member of HSP70 family of chaperones called BIP or BIP, that means binding protein. The formation of disulfide bonds between the side chains of cysteine residues is an important aspect of protein folding. and assembly within the ear. These bonds do not form in the cytosol, which is characterized by a reducing environment that maintains cystine residues in their reduced state. In the ER, however, an oxidizing environment promotes disulfide bond formation. And this is also facilitated by the enzyme protein disulfide isomerase, which is located in the ER lumen. Then protein glycosylation occurs in the ear. It occurs in an ordered sequence. Afterwards, the proteins or the modified proteins have to be transported from the ear to the Golgi, and it is transported by a vesicle fusion, or it is called vesicular transport. The proteins and lipids are carried from the ear to Golgi in transport vesicles that bud from the membrane of the ear and then fuse with the membrane of the Golgi. Luminal ear proteins are taken up by the vesicles and released into the lumen of the Golgi. Membrane proteins maintain the same orientation in the Golgi as in the ER. The distinction between proteins exported from and those retained in the ear can be governed by targeting sequence, which actually decides which proteins will be retained in the ear and which will be transported to the Golgi. Many proteins are retained in the ear lumens as a result of the presence of a targeting sequence: lysine, aspartic acid, glutamine, and leucine in the single letter code at their C terminus. And after that, in the Golgi, some processing also occurs in an ordered sequence. After that, CLATHRIN coated vesicles are produced, which transport the proteins destined for lysosome. Actually, we know that lysosome will be the final cell organelle in the GERL system, and in this case, CLATHRIN coating maintains, or it takes an important role. Actually, proteins targeted for lysosomes are marked by mannose 6-phosphates, which bind to mannose 6-phosphate receptors in the trans-Golgi network. The mannose 6-phosphate receptors span the Golgi membrane and serve as binding sites for the cytosolic adaptins, which in turn bind CLATHRIN. CLATHRINs consist of 3 protein chains that associate with each other to form a basket-like lattice that distorts the membrane and drives the vesicle budding. And afterwards, this vesicle fuses with the endosome. We can see this in the next slide. Molecules are taken up from outside the cell in eukaryotic vesicles, which fuse with early endosomes. Membrane components are recycled as the early endosomes mature into late endosomes. Transport vesicles carrying acid hydrolases from the Golgi apparatus then fuse with the late endosomes, which mature into lysosomes as they acquire a full complement of lysosomal enzymes. The acid hydrolases dissociate from the mannose 6-phosphate receptors when the transport vesicles fuse with late endosomes, and the receptors are recycled to the Golgi apparatus. So, in this way, after the endocytosis, lysosome is produced, and we can say in a nutshell that at first the proteins are synthesized by the ribosome, then it is modified within the ER. After that, from ER, vesicular transport occurs to transport the proteins to the Golgi apparatus. Then some processing occurs within the lumen of the Golgi, and afterwards from Golgi the proteins are transported to produce the lysosome, which ultimately is responsible for the cellular digestion. In this way, the vesicular transport helps in the GERL formation. Thank you, and in the next class, we will discuss about the specific structure and functions of endoplasmic reticulum, Golgi apparatus,